Okay, should we start with a sound check? Can you hear me there in the back? Can you hear me there in the back? Excellent. Lovely to see you all here on a Friday with bad weather. Welcome. I always appreciate having someone I can actually talk to. I'm sure people learn from the videos, but it's not nearly so personal for me. There is a test Wednesday, in case you had lost track of that. There's 3 different ways of drawing on our resources that are scheduled. One was the review session that's been announced earlier. That's going to be conducted by Susan O'Donnell, and that's on Monday, 6 to 8 p.m. Angie has arranged a place on Sunday at Hardin Library, and this will be posted, has been emailed and can be posted as well. It's more of a study group. It's not a formal presentation at the beginning, as Susan did, I think, last time, and we'll do again. More of a nuts and bolts interactive. Here's how I got through 110. And although I announced that I'd extended my office hours, I have to switch them to the afternoon that Tuesday. I'm sorry I gave you information that had to be changed, but something has come up that Tuesday morning. So I will be available, but Tuesday afternoon rather than Tuesday morning. And of course, Carissa Carlson is available on a one-to-one contact, email, individual appointments, or whatever you find useful. Any other questions about test prep and so on? I believe you've been working out some study questions for them too, right? Have those been emailed or posted or? Okay, they will be shortly. The study questions that you collectively wrote will be available shortly through ICON, I suspect. So keep an eye out for that and for an email about that. Any other questions about carbohydrates before we move on? There was a comment someone pointed out on one of the slides in the carbohydrate chapter on the enzyme fructose 2, the compound fructose 2,6-bisphosphate and how it modulates phosphofructokinase 1. Can the person who told me about that tell me what the slide number was? Slide 1022. There's a typo in there that no one spoke up and missed a chance to get an extra bonus point. So slide 1022, it increases the affinity for fructose 2-phosphate, not 1-phosphate. And I don't know why oh, there it goes. All right. Remember, I would be thrilled to be corrected in class, so don't be shy. Fructose 6-phosphate, I'm sorry. Instead of being fructose 1-phosphate, the substrate is fructose 6-phosphate. The substrate for phosphofructokinase is fructose 6-phosphate. Maybe I should open that slide. All right, maybe I'll try and find that slide. This is clearly baffling people. If only this thing went faster. Okay, hang on. So slide 10 what's this again? 10 what? 22, okay. So. But seriously, I would really like to be corrected in class if you see mistakes. I do try to make them correct, but I'm human. I haven't yet found out how to avoid them entirely. Okay. Where is this? Okay. Apologize for the speed of the slide of the computer. Okay. Here, increases for fructose 6-phosphate, not fructose 1-phosphate. Does that make sense now? Since the substrate for the compound is fructose 6-phosphate, that's what you want to change the affinity for. So I will put that in red and Add that to the, oh, what's it doing? What's it doing? Stop, whatever you're doing, stop. Okay. Put this into bold, italic. Never seen a computer that had to count on its fingers before. Anyhow, let's park that for now. I know, I'm going to have to save and close this or it's going to drive us crazy because nothing will work. Okay, so this one, no, not that one. Okay. I'm sorry, it's just not being very friendly to us. I apologize for the time lost. Well, let's see if this will work now. Let's start thinking about the citric acid cycle. And I'll put the lights down. There we go. This is a cycle that's had many names in its lifetime. I may occasionally slip and call it the Krebs cycle. It's had others as well. But it's a really amazing pathway. And when you start to get a feeling for it and what it does, you'll start to appreciate what an incredibly powerful cycle this is. This is the first pathway where really the pathway itself can be called amphibolic. We spoke about glycolysis being catabolic, breaking things down, and gluconeogenesis was anabolic, building them up. Citric acid cycle is amphibolic. It's both. And we're going to start by thinking about it in its catabolic state, when it's oxidizing stuff, because that's extremely important. That's how most of the energy in the body gets processed. It's a way to take molecules and generate ATP. So it's the universal converter. And it can burn everything. It can burn sugar, it can burn fat, it can burn amino acids. So it's a very flexible furnace, if you like to think of it that way. But it has a completely different aspect when you think of it as an anabolic pathway. And when it's an anabolic pathway, it's providing lots of different kinds of building blocks. And we're going to see that it makes amino acids, it makes the carbon backbones that are needed for gluconeogenesis. You can use the energy of fat to make sugar. You can't use fat to make sugar. But you need carbon backbones, and they come from amino acids. And it makes the precursors for heme. And of course, heme is very important in things like hemoglobin and the cytochromes we'll be talking about and so on. So what makes it kind of an interesting pathway to think about is that it is cyclic. There isn't a beginning and an end like glycolysis. There's a circle, and a circle that you can go around, and you can come in at many different points. We're going to start by thinking of it at the principal entry point when it's used catabolically, but do be aware that it has multiple entry points, and we'll kind of bring that forward as we talk. Any questions to this point? Okay. So let's take a little look at where we're going with this. And as always, we try to have a visible, high-level map for you. We've talked so far about breaking sugar down to monomers and breaking the monomers down to 2 and 3 carbon intermediates. We've seen that for the carbohydrates. We're going to see that again for proteins and amino acids, for proteins and fats. But we've got some 2 and 3 carbon fragments, wherever we got them from. We're going to feed them into the citric acid cycle. And from that, we're going to be pumping out carbon dioxide. So this is one of the principal places that carbon dioxide is released in us. We're going to be producing reduced compounds, NADH and ubiquinone. So these reduced compounds are good electron carriers, and those electron carriers are going to be used to produce the actual energy. They'll be fed into oxidative phosphorylation. And so we're going to take a little look at seeing how that pathway accomplishes that. One of the major fuels for the citric acid cycle in people with a mixed diet is going to be pyruvate derived from carbohydrates. You remember we finished glycolysis and we had at the end of it a 3-carbon fragment, pyruvate. If we were working anaerobically, we'd be making lactate or ethanol or something like that out of it. But if you want to get the maximum energy out of it, you want to burn it more efficiently, that pyruvate will go to the citric acid cycle. But it can't go straight in. It has to be funneled in through a very particular and peculiar kind of complex. And it enters through this enzyme monster called pyruvate dehydrogenase. And if you look at the dimensions on this, this is 200 angstroms, or 20 nanometers across. So this is a big complex. And it has in this kind of rugby ball or soccer ball 3 different enzymes, each of which is present in multiple copies. And altogether, that complex can have between 60 and 140 polypeptides in it, depending on the species we're talking about. Now, what we want to accomplish is this. We want to feed in pyruvate, CoA, the carrier, and NAD+, the oxidized form, and get out of it acetyl-CoA, the 2-carbon fragment, release carbon dioxide, and an NADH. So this is the reduced form here. So we're going to create a high-energy bond in this acetyl-CoA, driving that reaction by releasing a carbon dioxide and capturing a bit of the energy as an NADH. Sounds like a big job, and let's take a look at how we do that. The first enzyme in the complex is also sometimes called pyruvate dehydrogenase, or if you don't want to get confused by having the name for the subunit and the enzyme be the same, you can call it pyruvate decarboxylase. And what is it going to do? It's going to decarboxylate pyruvate. So here's our pyruvate coming in. And in this case, we're going to see the contributions of a cofactor that actually occurs in many places. So it doesn't, this is again a situation where the enzyme alone can't accomplish it through just the side chains of amino acids. It has a cofactor bound to it that's derived from a vitamin. This is derived from thiamine. This is thiamine pyrophosphate. So you can see here a pyrophosphate ending to it. By the way, customer alert, there are places that will sell you thiamine pyrophosphate instead of thiamine, and they'll charge you about 100 times as much for it. Since your stomach is acid, unless you have a very serious medical problem, that pyrophosphate will be digested off, so you might as well just buy thiamine. I don't like people who use biochemical ignorance to make money off of suckers. Anyhow, the interesting part of thiamine is this end down here, and what it's going to do is it's going to draw on the electron cloud on the pyruvate in such a way that a bond is created to the thiamine from the 2-carbon fragment, and allowing electron rearrangements that will allow this carbon dioxide to come off. And the covalently bound intermediate will look like this. And so what we've done is we've set it up to undergo the rest of the reactions. One of the classic examples of thiamine as a prosthetic group, but it's present in many other enzymes as well. E2 is the second enzyme, and it's going to receive that 2-carbon fragment onto a sulfur atom. And so it also has a cofactor on it. Again, side chains aren't enough. It needs a cofactor. This cofactor is something called lipoic acid. It has, some sort of hydrocarbonish bits and then this very odd sulfur bit towards the end. Lipoic acid can principally be made by our bodies, but some people don't make quite enough of it. So lipoic acid is also available as a supplement, although it's not recognized as an official vitamin. And so the lipoamide down here is going to sort of pluck off the carbon there from the thiamine. And in the process, end up with another rearrangement here, so we end up with a carbonyl oxygen there, and now it's covalently bonded to the lipoamide, and the thiamine pyrophosphate's been regenerated. Interesting example of a proton coming in and going out, so that altogether it's not, there's no net or gain or loss. But the thiamine pyrophosphate's now ready to be used again, so E1 is back to business. E2 can do a very cute trick. If you think back to, if you look at the structure here, you can see how the lipoamide has the 2-carbon fragment on a sulfur with a carbonyl oxygen right there. That looks like it could be a high energy bond, and in fact it is. It is sufficiently high energy that it can be attached to a coenzyme A, so the transfer of that 2-carbon fragment to the sulfur there is energetically a reasonable thing to have happen. And when the 2-carbon fragment is attached, we now have acetyl-CoA. It can freely diffuse away, so it diffuses away to be done something with. We're going to talk about it being used in the citric acid cycle, but you can do other things with it too. But unfortunately, the lipoamide is still in the reduced state. So it was in an oxidized state when this all began. It's clearly in a reduced state now. To get everything back to the beginning, we're going to have to oxidize this and get rid of those electrons somehow. And that's what enzyme 3 does. Enzyme 3 regenerates the lipoamide. So enzyme 2 regenerated enzyme 1. Enzyme 3 regenerates enzyme 2. And what does it do? Enzyme 3 is going to take the electrons from the lipoamide and put them onto FADH2. So this is flavine adenine dinucleotide. This is usually an enzyme-bound cofactor. If you remember back to chapter 9, FAD had some structural similarities to NAD, but they were a bit different. They're both good electron carriers, but a little different in how they're used. And the FAD usually stays bound to the enzyme that it's on. But when the FAD plucks off the electrons, we've regenerated enzyme 2. It's now back in the oxidized form. And the enzyme then is going to transfer its electrons to an NAD+, and that regenerates the FAD. The NADH that's been created here is free to diffuse away. So now we've been able to create an acetyl-CoA that can diffuse away and be used. We've created an NADH that can diffuse away and be used, and a carbon dioxide that just goes out and has to be gotten rid of. This is a little picture of the FAD, and so you can see here that this part of it, the adenine dinucleotide part, is similar to NADH, but instead it has a riboflavin side chain. And if riboflavin sounds familiar, it's another B vitamin. And so you're starting to see how the B vitamins are cofactors for a wide variety of these enzymes involved in intermediary metabolism. So if we look at these reactions all together as one thing, remember we started talking off about this thing as a great big beach ball of many, many subunits. And although we've talked about the reactions so far individually, they're in fact transferred one after another down that complex so that they all happen in a very smooth way. So we have pyruvate coming in, and at the first enzyme complex, it becomes covalently attached to the thiamine, releasing the CO2. The 2-carbon fragment on enzyme 1 is transferred to the lipoamide, so that you have a covalent bond now on enzyme 2, but you've regenerated enzyme 1. And enzyme 2 needs to pass off the 2-carbon fragment to CoA to make acetyl-CoA. It's still oxidized, and so to reduce it back to this, we transfer electrons onto the FAD of the enzyme, which transfers them in turn to an NAD+, which then is free to go away. So you can see it's kind of like a bucket brigade, you know, handing things one down after another. There's also some thought that by having all 3 of these enzymes on one complex that you increase the efficiency of it. There's a whole little subculture of biochemists who think about channeling. If you were to build this thing up, it looks something like this. You have here the different combinations of them. So here's lipoamide reductase, transacetylase. Here's the hydrolipoyl dehydrogenase. Here's the pyruvate decarboxylase. And if you pack the whole thing together, then you've got all those guys touching each other in a complex that allows for efficient reactions and regenerating all those vitamin cofactors. Any questions so far? Okay. All right. So we've taken glucose down to pyruvate through glycolysis. We've let pyruvate dehydrogenase turn the pyruvate into acetyl-CoA. Now we're ready to face the citric acid cycle. So what's going to go on? I think one thing that is immediately obvious looking at this is it is a cycle. It is not a linear pathway. And we could begin at many places, but since right now we're starting by thinking about oxidizing pyruvate, we'll start where pyruvate comes in the form of acetyl-CoA. So this is acetyl-CoA, the principal fuel for the citric acid cycle. In this case, it came from pyruvate, could have come from other things. In this case, it came from pyruvate. So we feed in an acetyl-CoA, and here is a 4-carbon molecule, oxaloacetate. It's got 2 acid groups and a carbonyl oxygen there. So those 2 carbons and 4 carbons together make this 6-carbon molecule citrate with 3 carboxylic acids and 6 carbons altogether. The fact that this has 3 carboxylic acid groups, as does this one, is why this is sometimes called the tricarboxylic acid cycle. That's another name you may see it by, the TCA cycle. So this is an enzyme cycle with many names. Citrate is a symmetrical molecule. You can see that the alcohol and the carboxylic acid are both plunked onto that middle carbon. The next step in the reaction is a conatase that is going to do a small rearrangement to move that hydroxyl down to that carbon. And that becomes isocitrate. Isocitrate because it's all the same groups, it's all the same number of atoms, but it's arranged a little differently, like isoleucine and leucine. Probably, as you can guess, a not very complicated reaction, and it's a reversible one. Isocitrate is then the substrate for the next step, which is alpha-ketoglutarate, is isocitrate dehydrogenase. So the name says isocitrate dehydrogenase. It's going to create a reduced compound. Its substrate is isocitrate. So what it does is it takes isocitrate, it releases another NADH here, and its product is alpha-ketoglutarate. Let's take a look at the structure of that. So it's a 5-carbon molecule, 2 carboxylic acids, and it's alpha-keto. So on the alpha carbon, a carbon 1 away from the carboxylic acid, it's got a keto group. So the name tells you what it is. This is clearly an oxidation step because we're releasing a reduced NADH. It's also a place where we've lost one of the carbons in isocitrate. Not the carbon we put in at the top. We'll talk about that later. But we've lost a carbon. And the fact that there's a carbon lost there is one of the things that helps to drive that reaction. Alpha-ketoglutarate is the substrate for the next reaction. Alpha-ketoglutarate dehydrogenase. Again, a clue, a redox reaction. Alpha-ketoglutarate is the starting material. And so we again see a NADH released and a carbon dioxide released. But in this case, we feed in also a CoA molecule and have now a succinyl-CoA. 4-carbon side chain attached to the CoA group. A little bit of an odd kind of a CoA compound. Not our usual shape, but we see the usual carbon with a keto group there and the 4 carbons there. Now, you've probably started to hear me say enough that can be equivalent in energy to a high-energy phosphate bond. We see an example of a substrate-level phosphorylation here. The succinyl-CoA has enough energy in it to drive the production of a GTP molecule. And when that happens, it releases the CoA and the molecule succinate. Succinate's a nice little symmetrical molecule with carboxylic acids at both ends. It can be oxidized even further. You can see it's still got some hydrogens in there, so it can be oxidized some. In this case, it's succinate dehydrogenase, and it's going to reduce the Q compound instead of NAD. So we're going to have reduced Q here. When we've reduced the succinate, we have fumarate, so 4 carbons. You can see that double bond in there because it's been partially oxidized. A hydration reaction across that is going to give us malate, and that's the substrate for our last dehydrogenase, which is going to produce another molecule of NAD and get us back to oxaloacetate. So it's a cyclic pathway. There's oxidation going on at several steps. Those electrons are captured in a useful form, and it gives us high-energy phosphate there. A couple of decarboxylations help to drive it in that direction, in the forward direction. Let's take a little look at some of those reactions in a little more detail. The first step is the one we mentioned here, where you have the 2-carbon fragment on acetyl-CoA and the 4-carbon oxaloacetate. And we're going to smoosh those together to make citrate, symmetrical citrate. And they've handily marked the new atoms in the citrate from the acetyl group as red. So you can kind of keep track of them as we go through. Releases free CoA, and because there is a thioester bond broken, it has a very strong negative delta G. So that's a forward reaction, one-way street, and a very major change in conformation when you look at what the enzyme does. So this is in the open state before things have bound to it, and here it is with the substrates bound. So kind of like, if you want to play enzyme tonight. I'm not going to go there. I'm not going to go there. I'm obviously getting tired. The next enzyme in the citric acid cycle is a conatase, and it has this rather odd name. Most of the enzymes we've talked about have very simple, meaningful names. This one's called a conatase because its intermediate is a conatate. And a conatate provides sort of a stepping stone in between the two forms of citrate. And as you can imagine, this is energetically equivalent. There's really nothing much to be distinguished between those two forms. You would think, given that it's starting from a symmetrical beginning point, that you'd end up with a mixture of things at the end, but we don't. And the fact that we don't get a mixture at the end puzzled biochemists for quite a while until something important was learned about the way enzymes work. So I'll just say there's a gun hanging on the wall and it will go off before the third act. Third enzyme, isocitrate dehydrogenase. I'm sure you're starting to get the sense that whenever you see a dehydrogenase, somewhere in there, there'll be an electron carrier playing a role. In this case, it's NAD+. Looking at this in a little more detail, so you can see here the place where the action is going to be, so to speak, and the carbon here that has an alcohol is going to end up as having a carbonyl oxygen. Clearly, that carbon is more oxidized than it was before. And that, normally speaking, making NADH is going to cost a certain amount of energy. In this case, that's driven by losing the carbon dioxide. And now we have alpha-ketoglutarate. Without the keto part, it'd be glutaric acid. This is alpha-ketoglutarate. And we're going to see that glutarate is related to glutamate and glutamine. So it's not a random name. So, fourth enzyme, alpha-ketoglutarate dehydrogenase. Another example of an oxidative decarboxylation. This is the third time we've seen that happen, right? Because we saw that in pyruvate dehydrogenase and the isocitrate dehydrogenase. Again, the NAD+ reduced to NADH. And when we look along the molecule, we can see that this carbon is going to be released. And in the process, that carbon can be considered to be relatively oxidized compared to there. If you think about what's going on here, there are some structural similarities or analogies to what had to happen at pyruvate dehydrogenase. So in the case of pyruvate dehydrogenase, you fed in a compound, you threw away a carbon dioxide but made a CoA compound. and some NADH. And if you look at this, you've got the carbon compound, you're throwing away a CO2, and you end up with a shorter CoA compound. So there's a lot of chemical analogies to the reaction at pyruvate dehydrogenase. And in fact, the enzyme complex for alpha-ketoglutarate resembles pyruvate dehydrogenase quite a bit, the kinds of reactions it has to go through, the cofactors, and so on. So the strategy was not is not radically different. It's using the same general approach. Now, one of the things that's kind of interesting and tricky about the citric acid cycle is that for every acetyl-CoA you put in, you're going to throw away 2 carbon dioxides. And a thoughtful person might ask, where did those carbon dioxides come from? And the interesting thing is they didn't come from the acetyl-CoA you just fed in. And if we follow these for a little bit, you'll see why. So here we have the oxaloacetate in gray and the acetyl-CoA in red. It may not be quite as dramatic on your handouts, but it is colored in your textbook. So let's see, that's 16. How much contrast? Oh yeah, there's plenty of contrast. So when we make the citrate, you have this nice symmetrical molecule. Isocitrate, asymmetry doesn't show up, the alcohol's down there, but that's where the carbons are that you put in. And we're going to see why that didn't get scrambled when we get a little further along. You throw away the first carbon, but it comes from that side piece there, so to speak. And we throw away a second carbon, and we nibbled it off the end. So we have still the 2 carbons we put in. We threw away 2 different carbons. And the succinyl-CoA compound goes on to reactions in here, in the rest of the cycle, that do scramble the symmetry. And so after it gets past this point, the carbons start to get randomly mixed together, so to speak, and eventually you do lose the carbons you put in as acetyl-CoA, but you don't lose them on the first cycle. So we have our succinyl-CoA, high energy bond in this CoA compound. The thioester is a very strong candidate for doing things. And an inorganic phosphate. This is, again, one of the few places where we see an inorganic phosphate being used to make a high energy bond. And what's kind of cute is the intermediate is a succinyl phosphate. So you can see that that would be energetically equivalent, that we've simply put the phosphate where the CoA compound was. The enzyme is named for the backwards reaction. So in other words, the name of the enzyme is succinyl-CoA synthetase, as if you're making succinyl-CoA out of succinate and GTP and CoA. But in the cell, it doesn't go that direction. It's named for the backwards reaction. This is another example of a phosphohistidine. I don't know if you remember back in glycolysis where there was a phosphohistidine that allowed us to store a high-energy phosphate during the reaction. So we have, and this is just showing you how that trick happens. So the succinyl phosphate transfers its phosphate to the histidine. That releases a succinate, and the phosphohistidine can then transfer its phosphate to the GDP. And the succinate is available now for the rest of the reaction. So, these last 3 reactions to complete the cycle have as one of their functions recovering oxaloacetate. If we didn't get back to oxaloacetate somehow, the cycle would come to a stop because making citrate requires acetyl-CoA and oxaloacetate. So part of what we're doing is making sure that by the time we get there, that this compound will be sufficiently oxidized to be an oxaloacetate. That's one way to think about it. The other is we're extracting the last bit of energy out of the compounds. Succinate dehydrogenase takes succinate and is a dehydrogenase, takes electrons off. But in this case, it puts it onto an FAD rather than an NADH. Enzyme bound like the previous FAD. And here's your fumarate with a double bond in the middle. This is a energetically equivalent reaction. Now, an interesting thing about this enzyme, succinate dehydrogenase, is it is the only enzyme of the citric acid cycle that's embedded in the inner mitochondrial membrane. So it's actually in the membrane itself. All the rest of the reactions are happening in the mitochondrial matrix. But because FADH is an enzyme-bound compound, you need to have it close to where it's going to be doing stuff. And in this case, those electrons are going to be funneled into oxidative phosphorylation. And because this enzyme is embedded in the membrane, it transfers its electrons to ubiquinone, often called just plain old Q. And that regenerates the enzyme for the next round of activity. So fumarate has this double bond. If we add a water molecule across it, you can see we now have an alcohol there and a hydrogen there. Energetically equivalent because this carbon is relatively reduced and relatively oxidized, so no net gain. And as you can imagine, it's a fairly reversible reaction. This reaction we will see again. We will see this again. So although it may not look very thrilling on the face of it, it will be an important one later on. Malate dehydrogenase, again, electrons are going somewhere, is going to oxidize the malate and reduce NAD+. So here is the carbon that's going to get completely oxidized, or be more oxidized at the end of it. The electrons from here are transferred to the NAD+, and we now have a carbonyl oxygen there. And that's oxyl acetate again, back where we were. So here we have it, an alpha keto carboxylic acid, alpha keto acid, if you want to think of it that way, and more NADH. This is not a terrifically favorable reaction. Its delta G is plus 30 kilojoules. That's not a very favorable reaction. But the citrate synthase reaction is really irreversible, and so it pulls this forward by withdrawing the citrate as it's available. So one way you can think of the citric acid cycle is that it's a catalytic cycle. That is, for a fixed amount of intermediates, you can pump through it many copies or many molecules of acetyl-CoA. So you don't have to have a separate Krebs cycle for each molecule. You can reuse it many times. And here's sort of a summary of what's going to be going on. In the case of glucose, you got some NADH during glycolysis, but you got 2 pyruvates out of it. We made the pyruvates into acetyl-CoA, yielding some NADH there. But clearly, the big work is coming in the citric acid cycle itself. Here's 6 NADH, 2 ubiquinones, and 2 GTP per glucose that you're feeding in. So this is balancing it for a glucose molecule, not just for a pyruvate. And the 4 of the carbons of the glucose came out in the citric acid cycle. 2 of them came out at the pyruvate dehydrogenase step. So clearly, a big chunk of the energy in glucose is coming out through the citric acid cycle. And we can see this dramatically because if you increase the amount of the intermediates in the citric acid cycle, it has a larger increase in activity than you would just predict from that size of intermediate because it can be used over and over again. Brings home the relative energetic efficiency of anaerobic and aerobic activity. So in the absence of oxygen, glucose is going to give you 2 ATP because those NADHs can't be used for energy production. You haven't got any oxygen to put the electrons on. They're going to have to be, one of these lactate exchanges or alcohol or whatever. So in the absence of oxygen, you get 2 ATP. In the presence of oxygen, you can get about 38. So far more effective a way to oxidize or extract energy. This is reminding you of the review hours. And I didn't see this to change it. I'm sorry, I forgot that this was in here. Please change that to 1:30 to 4 in the afternoon. And I will do that after class. given how slowly this works, I won't ask you to stop while I do it. All right. So something as central as the citric acid cycle cannot just be allowed to just wander around on its own. It needs a measure of control. If you've ever seen the huge wall charts of metabolism, and my husband always says I should start this class by saying, you need to learn all this semester. I've always told him I'm never going to do that. But if you look at those wall charts, embedded in the center often is the citric acid cycle because it connects to so many pathways. And so it's really important that it not just be waffling around on its own, so to speak. And so if we look at the pathway, it shouldn't be at all surprising that we see the regulation at the irreversible steps. Now, it doesn't make any sense to talk about a committed step with a citric acid cycle. It's a cycle. So you can't really speak of a committed step. But you can speak of irreversible steps, and all three of the irreversible steps have a measure of regulation. And if we take a look at what kind of regulation there is, a lot of it makes sense in terms of energy status. So if we look at citrate synthase, there's simple product inhibition, where citrate itself can go back and inhibit it. NADH can inhibit it. And actually, interestingly, succinyl-CoA can inhibit it. So that's like talking from halfway across the cycle. So the citrate synthase is getting information from several places about whether things are accumulating or not. Isocitrate dehydrogenase is another irreversible step. Again, NADH is a regulator. ADP is a regulator. That's a positive regulator. If ADP is accumulating, you're short of energy. Good idea to crank it up a bit. And calcium is a positive regulator. And that's because of reasons to do with the way mitochondria are put together. And we can talk about that later if you like. And the third regulated step is the third irreversible step, which is the alpha-ketoglutarate dehydrogenase. Again, a place where there's a decarboxylation. NADH is again a regulator for it. Again, we have feedback inhibition where the product is inhibiting it, as we saw up at the citrate synthase. Calcium is again a positive effector. So kind of a network of regulation. The NADH is acting at all three steps, which makes a lot of sense. It's a thoughtful way or a sensible way to regulate this thing because the NADH is a common output of the whole pathway. And these things are all good measures of energy status. If you have a lot of NADH around, you're not burning it as fast as you could, and so there's no point in making more of it. Whereas ADP is a good sign that you're short of energy. So, the citric acid cycle is an amazing thing. It is such a beautiful, complete pathway that people have asked the question, how on earth could something like that ever get started? If you need the whole pathway to get it done, how could you ever begin on it? And we interestingly have a clue to what might have happened because there's at least one very major group of organisms that does not have a functioning citric acid cycle. And that is the group of the blue-green algae or the cyanobacteria. Cyanobacteria don't have alpha-ketoglutarate. Now, cyanobacteria are very independent organisms, and they really kind of make their own way through the world. So if anybody was going to do without a full cycle, it's not too surprising that they're it. But if they don't have alpha-ketoglutarate, why do they have the rest of it? And the answer is, they use the first part, or the part downstream of citrate, if you like, for oxidation. So these things are, in fact, usable for oxidizing things. And you're going to get some NADH out there. They use the rest of the cycle in this direction as a way to make compounds for biosynthesis. And so going back down that way is a reduction pathway, and especially the succinate at the end is a compound that can be used to make heme. These other compounds like oxaloacetate can be used to make amino acids. So there's utility to all the reactions in the absence of alpha-ketoglutarate dehydrogenase. And that one enzyme missing suggests ways that the completed cycle might have come about. I tried to emphasize how many similarities there are between alpha-ketoglutarate dehydrogenase, right in here, and pyruvate dehydrogenase that made the acetyl-CoA. That if you go back and take a look at those things, you're going to see you feed in, odd number of carbons, you release a carbon dioxide in NADH, and you're making a CoA compound. And it turns out that you could easily imagine the evolution of an alpha-ketoglutarate dehydrogenase from a pyruvate dehydrogenase because of the similarities. You just need to make a slightly bigger binding site for the substrate. The business end is the same. So all you need to do is make a change that makes that pocket big enough to hold the alpha-ketoglutarate. So an interesting idea of how something that looks like it would have to be all there to be useful, in fact, can be useful in pieces. And let's haul a break there and have a good weekend. Thank you.